In this video, we will discuss about the plant tissue culture. Introduction to plant tissue culture. As you know that plant tissue culture is also known as micropropagation, or it is also called in vitro culture, and can be defined the growing of plant cells, tissues, organs, seeds, or other plant parts in a sterile environment on a nutrient medium is known as plant tissue culture, or it is also known as micropropagation or in vitro culture. In vitro means outside the body of living organism in laboratory condition, and in vivo mean within the body of living organism. So you know that plant tissue culture, for the first time, this process and technique is discovered by a German botanist Gottlieb Haber-Lehnt in 1902. That's why it is also known as father of plant tissue culture. The plant tissue culture cells, each and every plant cells are totipotents. Totipotent means it is the ability of each and every cells of the plant that it can be converted or regenerate the whole plants. This property of the plant cell is known as totipotency, and the cells are known as totipotent. Another property which is present in the plant is known as plasticity. Ability of plant to survive varying conditions by changing their metabolism, growth and development. It is known as plasticity. Plasticity have the ability to regenerate lost parts, ability to begin cell division from tissue of the plant, ability to regenerate any tissue or organ from any other type of cell. So plasticity and totipotency, it is the ability of the plant cells, not present in animal cells. These are various types of tissue culture apparatus, cell culture hood, which is also known as sterile chamber. It is also known as laminar airflow cabinet or biosafety cabinet or biotron. So various other names are also used for cell culture hood. So don't be confused. Cell culture hood, laminar airflow cabinet, biosafety cabinet and biotron or sterile room or chambers is the same thing. Incubator, it is known as a culture room, water bath, centrifuge, refrigerator, cell counter or hemocytometer, which is used for counting of the cell. Microscope, as you know that it is seen for those things which cannot be seen with our naked eyes. Cryo storage container, cryo mean snow or ice, and storage container with the help of which we preserve various types of material. Autoclave and sterilizer, used for killing of various types of pathogen or contaminants. Glassware, test tube, beakers, and conical flasks are used in the laboratory of plant tissue culture or for techniques of plant tissue culture. These are the laminar flow cabinet, which is also known as sterile chambers or culture chambers. Yes, these are known as incubator, and these are the machine known as autoclave for the removal of pathogen or contaminants from various types of plant material which is used in plant tissue culture. So the important thing in plant tissue culture is media preparation. As you know that soil is the natural medium for the plant growth, which is supposed to be very complex for 18 explants. So explants are grown in artificial medium. There are two types of media, semi-solids as well as liquid media, and it depends upon various types of plant species. Each and every plant species, they require different types of medium, so different types of medium can be prepared. Either they may be semi-solid or liquid. Media of a plant basically composed of inorganic salt plus organic nutrients, vitamins, plant hormone, either various types of plant hormone, auxin, gibberellin, cytokinin, brassinosteroid, and artificial as well as natural form they are used. Ethylene diamine tetraacetate and carbon source are used within the medium. These are various types of chemical, organic, inorganic, plant growth regulators, etc., which is used in media preparation. Now we'll go to the procedures of plant tissue culture, or we can say that 5 steps in plant tissue culture. The first step is selection of explant, then second step is surface sterilization, third step is inoculation, and fourth step is shooting and rooting or multiplication, and 5 number step is hardening. So the first step in plant tissue culture or procedure in plant tissue culture is selection of explant. The initial plant part of which is used to develop plant tissue culture is called explant, or any starting material of plants which is used for the process of plant tissue culture is called explant. It may be a complete organ, for example seed, leaf, stem, root, petiole, bud, or it may be protoplast, or it may be a piece of tissue. Usually the younger, the less differentiated tissues are generally used. It may be apical bud, meristematic tissues which have the ability to divide and redivide. The second step in plant tissue culture is surface sterilization. So plant tissue culture is performed under aseptic conditions. So removal of various types of microbial contaminants, pathogen, virus, bacteria, and fungi is known as surface sterilization. So surface sterilization of explant starting material takes place in chemical solution, especially the sodium or calcium hypochlorite or mercury chloride. 1% solution is used for surface sterilization. All the material, for example, vessel, instrument, medium, plant material, which is used in plant tissue culture, will be free from microbes. The glassware should also be sterilized. The third step is inoculation. Inoculation means the transfer of explant into medium is known as inoculation. And this process takes place basically in laminar flow cabinet or in a sterile chamber. After placement of explant, test tube, beakers, and conical flask, etc. should be airtight. Glassware, test tube, beakers, and conical flask, etc. should shifted to incubator growth rooms. The fourth step is multiplication or development of callus. As you know that explant gave rise to a callus within the nutrient medium within the incubator, and the callus can be defined as unorganized mass of cells or mass of loosely arranged cell or undifferentiated mass of cells. So the callus should be shifted to the new media for the development of shoot and root. Balance of both auxin and cytokinin often produce callus. The callus may be subdivided a number of times. The fifth and the last step in the plant tissue culture is development of plantlets, or it is also known as hardening. As the callus grow, pieces are typically cut off and transferred to new media subculture to allow for growth, so it may be multiplied. Varying sugar concentration and low oxygen and high cytokinin ratio to form multiple shoot. So multiple shoot basically emerge from a culture root formation. The shoots are transferred to a growth medium with relatively higher oxygen ratio. It produces root formation. Hardening means the root and shoots are potted up. It removed from test tube, beakers, or conical flask, and it is hardened up by gradually decreasing the humidity. This process is known as hardening. This is necessary as many young tissues culture plants have no waxy cuticle to prevent loss of water. So the plants, they should be deplusked and shifted from incubator to the greenhouse. And when they become hardened according to their environment, then from the greenhouse it is transferred to the open field. Tissue culture techniques based on explants. There are various types of plant tissue culture. For example, meristem culture, protoplast culture, cell suspension culture, bud culture, organ culture, callus culture, embryo culture, as well as a seed culture. We have to start callus culture. As I have told that callus is unorganized and undifferentiated mass of actively dividing cells. Darkness and solid medium gelled by agar stimulate callus formation. The medium contains the oxine and benzyl aminofurin, which is a type of cytokinin. Both are growth regulator hormone. It produces the callus. This stimulates cell division in explant, and callus is obtained within 2 to 3 weeks. What is callus development? Callus can also be naturally developed on a plant. As a result of a wound, or it may also be produced artificially within the nutrient medium by the process of plant tissue culture. Callus culture, as I have told, that equimolar amount of oxygen and cytokinin basically stimulate as well as darkness stimulate the cell division, and it produce undifferentiated mass of cells, which is known as a callus. Callus culture can also be cut up into small pieces, and further plantlet or multiplication process also increases. Or callus culture may also be converted into cell suspension culture when callus is agitated or with the help of a machine. So various types of cells or group of cells are removed from the callus, and then it can be used for cell suspension culture. A cell suspension culture, it is the another techniques used in plant tissue culture. Cell suspension culture basically develop from the callus when it is placed in a liquid medium. When callus is placed in liquid medium and it is agitated, disturbed, so single cells or some group of cells are released. Under these cells, some cell continue to grow and divide, eventually producing a cell suspension culture. Repeated subculturing into fresh medium. Cell suspension culture should be repeated into pressure medium. Cell suspension culture are useful to obtain some drug compound which are generally obtained from specific parts of adult plants. The cell suspension culture produce the same chemical as the entire plants from the cell suspension culture. For example, Cinchona legeriana cells can be grown within the cell suspension culture to produce the important drug quinine, which is used as anti-malaria. And those of Digitalis lentina, it produces a digitoxin, which is known as heart tonic, and give to the heart patient. These are basically the procedure. First, we have to select the plant, then surface sterilization, then callus formation, then callus should be agitated, disturbed, group of cells and tissues is removed from the callus, which is known as cell suspension, and these cells which is derived from a specific plant species, it will produce specific chemical. For example, quinine from cinchona and digitoxin from digitalis lentina. The third one is protoplast culture. First of all, what is protoplast? The plant cell in which the cell wall is removed is known as protoplast. Is known as protoplast. Our protoplast are plant cell when the cell wall is removed commonly isolated. The protoplast commonly isolated from leaf mesophyll cells are cell suspension culture. Protoplast culture can also used to develop whole plant by organogenesis. This single cell protoplast they regenerate the cell wall and it convert into whole plants by the process of organogenesis or somatic embryogenesis, because the cell is taken is a somatic cell and when they convert it into embryo and then embryo is converted into whole plants, this process is known as somatic embryogenesis. So synthetic somatic embryo that are developed from somatic cells is known as somatic embryogenesis. Genetic variation can also be produced in these somatic embryo. They are exposed to physical or chemical mutagens, and you know that physical or chemical mutagens are any structures or any material which is responsible to produce mutations. So this type of variations which is produced by physical or chemical mutagens within the somatic embryos is known as somaclonal variations. So protoclast act as ideal target for transformation by a variety of means. So various types of useful characteristics are produced in plants so according to the needs of human beings. So these are basically the protoplast culture. These are the plants in which we produce various types of radiation, which is known as physical mutagens. So protoplast can be isolated because the plant cells having cellulase enzyme, it will digest the cell wall. Naked protoplast cells are produced. Then, as you know that due to the radiations, mutation is produced within the DNA. We culture the protoplast, they convert them into callus and providing various types of plant growth regulator plantlets are produced. So protoplast culture is very important method for somaclonal variation as well as for somatic embryogenesis. The fourth one is meristem culture. As you know that meristems are actively dividing cells present at the tips of root and shoot, and they are used as explant, and this type of culture is known as meristem culture. So meristem culture is mostly used to obtain virus or parasite free plant because the whole plant may be infected by virus, bacteria or fungi, but meristems are devoid of pathogen due to the absence of vascular system in them. As you know that the meristems are those tissues which is present in the apex of shoots and root having no vascular bundle. Due to this, the pathogen will also absent and they are used as a explant. And this type of culture is known as meristem culture. These are basically the procedure, as I told that these are active dividing cell which is present at the tips of roots and shoot. These tips should be taken, sterilized, and then transferred to the nutrient medium. Plantlet are produced and these plantlet are free from various types of pathogen, virus, bacteria and fungi. So these are basically the procedure which is known as meristem culture. The fifth and last one is known as anther culture. It is also known as pollen culture or microspore culture. It is an artificial technique by which the developing anther are cut at critical stage aseptically from unopened flower bud and or cultured on a nutrient medium where the microspores within the culture enter develop into callus culture or embryo that give rise to haploid plants. Formation of haploid plants either through organogenesis or embryogenesis is known as anther culture. The first report of haploid tissue from anther culture was in 1964 in pollen grain of Datura, family Solanaceae by Maheshwari. There are production of haploid reported in about 250 species in Solanaceae family. Crucifery, Gramineae are most common. So anther in pollen culture is referred to as androgenesis, the male gametophyte, the formation of male gametophyte. Microspore are immature pollen produce haploid plants. These haploid plants can also be converted into diploid plants by the application of a chemical known as colchicine. These are basically the procedure. You know that anther is present within the stamen, and stamen is the male reproductive part of a flower from which the anther are obtained. Anthers they will be shifted to the nutrient medium where they produce the callus and or they produce the embryo. From the embryo it produce the haploid plants. And if we apply chemicals on these haploid plantlet or these embryo or callus, by the application of colchicine, it can be converted into diploid plants. Then, according to the characteristics of their production and their yield are considered. If they are fruitful, then they are used to the for various types of experiments. So, and these are known as anther or microspore culture. If you like the video, please like, subscribe, share, and watch.